to the invited lecturer, Professor Tomasz Guzik, Molecular Basis of the Effects of Environmental Pollution and the Cardiovascular System. The floor is yours.
Thank you very much, and I understood the suggestion about the break very well, so I will do my best to not delay that important aspect of our health. So as has been discussed, what I will try to do is maybe summarize a little bit the various aspects of the mechanistic effects of air pollutants, and I will focus, there was just a question just a second ago that we should maybe identify which pollutant within the air pollution actually plays the greatest role. And in order to be able to actually focus on something, I decided to focus on PM 2.5 molecules because they seem to have the largest amount of data available. And but when we speak about environmental pollution, I always want to start with reminding you something that has already come up with in the previous sessions, that we need to look at the environmental factors in concert because they all interact with each other, they all depend on each other, and soil pollution, water pollution should not be disregarded when thinking about various health effects that we can have. And of course, we've already heard the global burden of disease data, and this is an image taken from a recent review from Thomas Munzel and Andreas Daiber that has just been published in Cardiovascular Research, which is an expert statement emphasizing that air pollution and high blood pressure are taking the two top places. So that's why I think looking at their interaction during this conference is particularly valuable and important. Of course, you know, all the numbers have already been mentioned, and but I think that we need to emphasize that cardiovascular mortality is actually the top sort of reason that has been attributed to these numbers of exposure of air pollution worldwide. And that's why the link between cardiovascular system and the air pollution is so important for us. I think when we think about the molecules, and I said I will focus on air pollution and on PM 2.5 in particular, because there is, in my opinion, the greatest amount of evidence related to that, we need to realize that PM 2.5 is a very complex molecule that contains a lot of factors, a lot of chemicals, and a lot of organic compounds. And I will show you, for example, studies coming from Edinburgh that are showing that there is actually a difference in response to various types of particles that the animals in experimental settings are exposed to or cells are exposed to. The first question, of course, is how they enter into the cardiovascular system and which parts of cardiovascular system these particles can reach. And because there are various ways that these particles will be able to affect cardiovascular health. One is, of course, direct presence of these particles in the vessels and in the heart. The other is inducing pulmonary responses that will lead to systemic inflammatory disease, and we know very well and agree now that increased CRP, increased inflammatory markers are risk factors for cardiovascular disease development. And then there is, of course, the brain effect. So it is possible that the effect may be direct, may be indirect, or may be in fact combined. So there is quite a lot of evidence, and we already heard some evidence before in the earlier session, that there is actually uptake and appearance of these molecules in the vascular system. And there are different ways that different parts of the cardiovascular system will be affected. The most obvious one is when a foreign body is reaching immune system, it's being ingested by macrophages. And once the PM 2.5 is ingested and enters macrophage, it will change its biology. It will be partially treated, as every external molecule that macrophage would encounter. So if we give latex to a macrophage, it starts throwing huge amounts of cytokines and shows enormous inflammatory activation. So with macrophages, it's easy to imagine, but other methods such as endocytosis, even direct penetration of particularly ultrafine particles are possible within other cells within the within the cardiovascular system, including endothelial cells and including the smooth muscle cells. But the very interesting study that I want to draw your attention to, published a few years ago from Edinburgh, from Nick Mills and Mark Miller's group, have exposed people and animals to gold particle inhalation. And gold particles were sized to correspond to the size of the particular matter. And then they did this because then they can really look at the presence of gold in various areas of vasculature. So they did this before people underwent surgery, and they obtained blood vessels. And they saw that in blood, in liver, in aortic arch, in descending aorta, these gold particles were indeed appearing, and they could image them using confocal microscopy, and were able to measure their presence in various ways. And indeed, you know, the other studies that are more experimental, this was a very nice study because it was done in both humans and in animals in vivo, but already in vitro exposure of endothelial cells, for example, to these particles results in their entering the cell through endocytosis. And I think that is a very important aspect, because if a molecule like this, or a particle, is not a molecule, a particle like this, full of various organic and inorganic components will enter the biological system, it will unquestionably change its function and its behavior. And indeed, we see both on the clinical level and on molecular level these changes present. This sort of presence of these molecules can be very precisely monitored. We have here images from electron microscopy, confocal microscopy, fluorescent labeling, and X-ray identification of these cells entering into the cell. But this is on the right on your side is an image describing an experiment done by Mark Miller, and in which they compared the effect of just the sort of properly organically compounded molecule versus just a sort of an iron sort of a metal structure. And we can see, and this was a study of the effect on endothelial function. So this is for example, my lab, we always show it inversely, because this is a relaxation. So the higher the curve reaches is the higher relaxation, so better endothelial function. Very good in control, 100% relaxation, but impaired already by the metal, let's call it molecule, and truly impaired if the organic elements are absorbed on top of it. So it shows that very clearly that the properties of the molecule will have a huge effect on this fact. And this sort of links in part to this is a study that we performed in Krakow at the time where Krakow was particularly badly polluted, where we tried to answer the question, what is the relationship between the measures of pollution in the vicinity of people's inhabitants and the endothelial function that was measured on that particular day. We measured it a few times during the study on different days with different levels of pollution. We also calculated differences, but overall, all of the results were quite similar, that actually PM2.5 and PM10 appeared to be significantly correlated to the air pollution in the ambient air surrounding a place of where the person lived. While interestingly, the other pollutants were not as strongly associated. There were some trends. Of course, there is always a discussion of power for this study, but unquestionably, the particular matter seems to affect endothelial function to the greater extent. And indeed, there are studies published in the acute exposure of patients showing the impairment of endothelial function in response to acute exposure of an individual to PM2.5. And as you can see, FMD decreased up to even 3%. And if you measure FMD, you are aware that 3% is not that little, within hours of PM2.5 exposure, which translates, all of these measures are related to nitric oxide bioavailability, coupling of nitric oxide synthase, or oxidative stress that Omar has already mentioned, and I will show you some data in a moment. When thinking what are the principal mechanisms, I would always start with actually the inflammatory mechanism probably and the oxidative mechanism as the two principal components or elements induced in the various vascular cells by particular matter exposure. And the first one I already briefly discussed is, appearance and ingestion of these molecules within a macrophage will change the biology of that cell and will potentially upregulate the CD66 molecule on its surface, which makes it more prone to become a foam cell, which would explain an increased susceptibility to development of atherosclerosis in animal models exposed chronically to PM2.5. The evidence on oxidative stress is coming from various experimental studies in both animals and in cell culture, showing that both surface metals and organic compounds on PM2.5 catalyze and stimulate ROS formation. There is mitochondrial dysfunction, there is antioxidant depletion, and as Omar has mentioned, there is a change in NADPH oxidase expression. And here we see the Western blot. In Western blotting, the thicker the band is, the more protein appears, and we can see that in the case of exposure to PM2.5, we have increased expression of both NOX1 protein and NOX4 proteins, which are key elements of free radical generation by vascular cells, such as smooth muscle cells and endothelial cells in both human and in animals. And in this study, the authors went on and showed, for example, that green tea was inhibiting this induction, but this opens a very important question, how we could modulate these aspects. And of course, there are a lot of studies, I do not show for the time's sake, looking at various biomarkers of potential oxidative stress. For the person who studies and measures free radical production using paramagnetic resonance, I always take these with a grain of sand because they are very indirect measures of oxidative stress, and they are affected by many other factors, including inflammation. One of the key elements, and Professor Stassen was discussing the left ventricular function, and for left ventricular function, something that is very important is myocardial energetics. And there are actually studies showing that particular matter, when interacting with cardiac myocytes, can actually change behavior of mitochondria. And mitochondria constitute about 30 to 40% of cardiac myocyte volume, indicating that if we change mitochondrial function, then we will actually significantly alter the efficiency, energetic efficiency of cardiac myocyte, which could explain various aspects of links between accelerated heart failure and air pollution. The activation of inflammatory cascades goes beyond just the ingestion by macrophages. There is evidence that these molecules, especially the organic compounds that are present there, may actually work by almost as a pattern recognition receptors. They can activate the inflammasome, and they can stimulate a whole range and cascades of inflammatory responses in response to this fact, leading to systemic inflammation and leading to local inflammation in both vessels and, of course, in the lung. They can also interact. They interact, as you see, with practically every blood vessel cell in the blood vessel wall. In smooth muscle cell, we also have evidence of something called phenotypic switching that accompanies atherosclerosis. This is a phenomenon in which smooth muscle cells which usually constricts and, for example, increases our blood pressure when it's needed, it starts actually producing bioactive compounds such as cytokines. It starts proliferating, it starts migrating and misbehaving in various ways. And we can see indeed that exposure of these cells in culture to PM particular matter 2.5 in particular is resulting in changes in reduction in contractile properties and increases of these synthetic properties of these cells. Other aspects of cardiovascular function are also affected. There are changes in ventricular arrhythmias that can develop through various triggers, as indicated by a number of studies, and of course, developed studies looking at the relationships with heart rate variability that find quite a lot of different potential explanations. The last 2 or 3 points or mechanisms that are affected, of course, have to be related to thrombosis, because thrombosis is the culprit of the acute events that might be and have been shown to be associated with rapid increases in air pollution. So indeed, PM2.5 interact also with platelets. So they can be, as mentioned, as they can be carried by the red blood cell, they can also be carried by a platelet. And when they are actually interacting with a platelet or interacting with red blood cell, they change the properties of that cell. And this is very important. For example, it's very interesting. I actually have not read this interesting paper that they attach to red blood cell, but we can imagine that if they attach to red blood cell, they will change mechanical properties of red blood cell. And they will make red blood cell, for example, it make more difficult for red blood cells to squeeze through our microvasculature, leading to microvascular dysfunction. And in platelets themselves, we see features of platelet activation and aggregation. We see increased also coagulation cascades, impaired fibrinolysis. So all of the elements that would alter our coagulation and thrombosis in adverse. And summarizing these, we can appreciate that a number of different biomarkers have been associated in various studies with exposures to PM2.5. Both cardiac injury markers have been used in many studies. Cardiac stress and overload features, inflammatory cytokines, all of them have been shown to change potentially in results to exposure to with PM2.5, which is summarized here with inflammatory being the most predominant one, but we can see a breadth of different effects shown on this slide. What is important is that there are certain groups of patients that may be particularly vulnerable because of interconnection of the activation mechanisms induced by PM2.5 that are also activated within these. And this includes elderly patients, children, people with pre-existing cardiovascular disease, or patients with metabolic disease. And we have performed a small study in Krakow, which again suffers from all the drawbacks that have been discussed, which is the reason it's published in scientific reports. And in fact, you know, when I was preparing this, I always start preparing a talk from searching Nature, and I look for papers in Nature, papers in Science. Really, there is very, very little evidence in Nature and Science on these because of the difficulties in methodological unequivocality that these journals are requiring. So anyway, this paper is in scientific reports, but what we did is we used the fact that Krakow is indeed one of the most polluted cities, or at that time it was, and we decided to compare it with Lublin, which is to some extent similar, apart from actually a noise exposure, which surprisingly is higher in Lublin than in Krakow. So we had an opposite effect, which we learned only later when we actually started tracking for noise exposure. But you can see that the overall sort of, in general, features, the cities are comparable, but they differ very, very significantly in air pollution levels, and these are averaged values, of course. And what we did is we recruited children from these cities. We recruited, adolescents, I would say, and we measured number of cardiovascular parameters, and I will only show blood pressure because we are here as a European hypertension. We also, of course, measured various biomarkers and features such as BMI that I will refer to briefly. But overall, in the whole population, we did not see a big difference between the cities in children properties related to blood pressure. But interestingly, when we split them into girls and boys, we could see that only in boys this effect was observed. And what was very interesting was that the city of inhabitants was actually interacting extremely strongly with obesity as a factor to modulate blood pressure. And then last two slides, I think, is the temporal relationships. We always, in experimental models, very often we look at acute exposures. And these have to be differentiated from chronic exposures that have been done in very few experimental studies. And the effects between acute and chronic exposure may differ. So we really need to have more chronic studies. Overall, we can summarize that acute effects include inflammatory increases of C-reactive protein, increase in fibrinogen, drop in flow-mediated dilation, so endothelial function, and features of sympathetic activation. While chronically, there are studies linking exposure to IMT increases, to LV mass, to features of reactive oxygen species in cardiac myocytes and mitochondrial dysfunction, and changes within DNA of cardiovascularly relevant genes. So I think that this concept and aspect of exposure needs to be taken into account when thinking about this fact. And then Professor Konduratska, in one of her last slides, she emphasized that indeed we have no real evidence to say that there is a threshold that is safe. There are thresholds that are indicated, but there is clearly a nonlinear relationship with steeper slope at lower concentrations. So I think that this needs to be taken into account. And there are some papers that are trying to sort of draw conclusions that are a little bit overstated, such as shown here in this box. But these things, I think we need to try to understand better in order to be able to draw. So I think that a myriad of different environmental exposures that we are yet to understand. In recent 2 years or 3 years, we suddenly started appreciating that plastic and microplastic exposure is a great risk factor. Although I have to say that my daughter at school, already 5 or 6 years ago, she came as a kid and she told me, "Dad, we eat the whole credit card once a month in plastic," which I thought was a joke, but it turns out it's not only we eat it, but we actually get that credit card into our blood vessels and the heart. But I think we know already quite a lot about these mechanistic aspects, and they are quite convincing to us that indeed the air pollutants have significant biological effects that would translate into the cardiovascular mortality and morbidity. But as already indicated, I think we need stronger evidence to this sort of diffuse some criticisms related to methodological approaches in these studies. I already referred to a huge discussion that we had related to one of the top cited papers in cardiovascular research that I have an honor to lead as a journal, and the discussion was hitting the notes that by showing these kind of papers, we are diminishing the importance of smoking, for example. I think we need to be very, very careful and important to project these messages very strongly, because I think that these sort of messages tend to undermine the public understanding of the importance of air pollution. And with the increased need for energy in the forthcoming years, I think we will be facing even greater crisis in air pollution. Thank you very much.
Thank you very much, Tomasz, for your brilliant, as usually, lecture. And I think that there is unique opportunity to ask an expert. So then I think we have a time for a short lecture, short questions with the accent on short, because it is before the lunch.
Thank you. Very nice presentations. Really appreciate. I just have a question, for example, on macrophage. Isn't it the role of the macrophage to pick up this external substance and tox it and to try to eliminate it, through foam cells? So my point is, when is it a normal reaction and when does it become a pathologic reaction?
I think overall is always with the issue with the dose.
Yeah.
I think if a macrophage is overwhelmed by exposure to these particles, and we have so many macrophages in the lung, that their first job is to pick this up.
It's a defense.
What happens is they start overproducing huge amounts of cytokines. What we are missing actually is controlled studies which are comparing the proper ingestion of PM2.5 versus for example, the study that was done by Miller and Mills, where they had the organic and non-organic version of the molecule, and see how the macrophage differentially responds. I have not seen such a controlled study, because I think that would answer in part your important question.
Because it's the same also for endothelial function. Endothelial function is not a fixed function. It depends on the local vascularization, what happens, temperature, whatever. And it has to be changing, because the NO is a local regulator of flow in organs and in different tissues. So how much of these changes are adaptative or inadaptative?
I think in regard to this, we always try to emphasize that there is a difference between physiological regulation and pathological dysfunction. So I think endothelial dysfunction is, I would argue, always pathological. But it is not physiological NO regulation of blood pressure, vasodilatation, and so on. These are two different processes, and I think it's the same between blood pressure regulation and hypertension. I think that sometimes people put equality between them, and we know very well that this is different mechanisms.
I'm sorry, it's not just for you, but for him, about macrophage. In our study, we were able to get imaging by electron microscopy, by which we have evidence that the micronanoplastics were present inside living macrophages within plaques. So, of course, macrophage, I agree with him, is a key factors in the diffusions in the bloodstream, at least, of let's say pollution, but in our case it was micronanoplastics. And this is a point that should be understood, because the point is, if the macrophage is a vector, a shuttle, or macrophage is something by which is used by our body to defend from.
I think it's the old chlamydia discussion that, the chlamydia was present in macrophage and was brought over to the plaque. But from the evidence of the presence, I agree. But the presence of these particles have been shown to change function of macrophage. And I have always this discussion with Charalambos Antoniades, who is putting forward this hypothesis that TNF-alpha and interferon production from smooth muscle cell is affecting fat outside. But we are arguing with him that, if you have one macrophage in that fat, it produces 1,000 more TNF-alpha than a smooth muscle cell will ever do. And if it has ingested the PM2.5 or microplastic molecule, it is not known that much what is the functional change, but I suppose these functionally are usually changed.
I think that the next time for the discussion will be during the lunch break.
Thank you very much.
I would
congratulations on this very nice and important conference.
I would like to invite everyone for a lunch break, and I hope we may start the next session, because we have a slightly delay at 10 past 2 p.m. 10 past 2 p.m.